guys, I am Tebosolo. Here we meet again. I am a biology and chemistry teacher. So right now we are going to deal with the grade 9 biology, whereby we are going to do the movement in and out of cells. So we are just going to concentrate on diffusion and osmosis. So here I have written the definition of diffusion and the definition of osmosis. So I would like to start with diffusion here. So diffusion here, the definition says, is the movement of particles from a region of high concentration to a region of a low concentration down the concentration gradient. So now diffusion can be demonstrated in some ways. If I take maybe a perfume or a body spray, I spray it here where I'm standing, the particles of the perfume are gonna fill up this room. So which means somebody at a corner or some people in this room, they can smell that perfume. So which means now the particles of that body spray, they have moved across this room. But then right now it will be diffusion in gases, because the particles of that perfume are going to travel through the gases. So it means now for people to smell that, the particles are going to travel across the room. But at some time, the smell is going to fade, which means the smell is now gone. So now, in liquids also diffusion can take place. In the laboratory, we have the beakers. We can take the beaker and then fill it with water. So after filling the beaker with water, then potassium permanganate can be poured inside the water. So when potassium permanganate is put inside the water here, what is going to happen is the particles of potassium permanganate are going to diffuse in the water without being stirred. Here we don't stir anything. We just let potassium permanganate to diffuse in the water. So what is going to happen is that these particles are going to diffuse inside the water. These particles are going to diffuse inside the water. They are going to diffuse inside the water up until the whole water now is purple, which means now that particles of potassium permanganate are now going to diffuse inside the water up until this water have changed the color. But remember, you don't stir anything. We will do it as a practical experiment so that you will see when the particles of potassium permanganate are diffusing in the water. You will see these particles will be rising inside the water and creating something like a cloud, changing the color of the water. So now, in our daily lives, diffusion takes place everywhere. For an example, we can talk of the process of photosynthesis, whereby plants are going to take in carbon dioxide and then they are going to give in oxygen. So which means that process is going to happen by diffusion. Carbon dioxide is going to diffuse inside the plant leaf and then they are going to give out oxygen. So this means even in human beings, people, they breathe in, they take in oxygen and then they are going to breathe out carbon dioxide. So this is what is going to happen in between the lungs, whereby carbon dioxide is going to diffuse into the women's bloodstream and then the person is going to breathe out and give out oxygen. So that is some few examples of diffusion in our daily lives. So now I would like us to pass on to osmosis. Osmosis, movement of water molecules from a region of higher water potential to a region of low water potential through a semi-permeable membrane. I would like to talk about the concept of high water potential. What do we mean when we say something has high water potential and something has low water potential? Here, suppose I have my two beakers and this is they are filled with water. So now I have pure water, which means the concentration of water molecules in pure water, they are high. So now, immediately, if I put something inside the water, for an example, I can put sugar or I can put salt. Immediately, if I put salt inside the water, if I add salt, it means now the water potential, high water potential, is now going to be reduced because I am going to put something inside that water. So immediately if I add salt, now I can make my water to become low water potential. If I put more salt and more salt and more salt, it means now I am making a concentrated solution. So the more I put the salt, the more the water potential is being reduced. So now suppose I have beaker A and I have a beaker B. Beaker A has water and salt, while beaker B is only pure water. So this means here in pure water, I have high water potential. Here, whereby now salt has been put inside the water, it means now the water potential in here is lower than in that container. So now, even in here, I can put salt. I can put salt. Here I have 2 particles of water. This is no longer pure water. It means now the water potential is not high. But comparing these 2 beakers, you can see that here I have 2 particles of salt. Here I have much particles, which means 4. So this means here the concentration of sugar is much than in that container. So this means now the water potential here, water potential is higher than in here. But then if I put more salt and more salt and more salt and more salt, now this means the salt particles in here, they are now plenty, which means now here the water potential here, now the water potential is lower than in the beaker solution A. So now we can demonstrate we can do another experiment showing what will happen when there is a semi-permeable membrane. Now I have my bowl here, and then we are saying this is now, we are going to demonstrate osmosis now using a semi-permeable membrane. So this is now our semi-permeable membrane, and then here I will have pure water. This will be pure water, and then here I will have a solution, water solution. I can use salt again. Here I have a solution, salt solution. I have a salt solution. So now the definition says movement of water molecules from a region of higher concentration to a region of lower concentration. So it means now here I have high water potential because this is now pure water. Here I have low water potential because now this is a solution. There are some sugar particles inside the water. So this means now water potential in here is lesser than on the other side. So this is what is going to happen. This is the beginning of our experiment. It means now water molecules are going to move inside the solution of salt and water, which means at the start, now water is going to move from a region of high water potential to a region of low water potential. So it means water is going to move from this side to this side through the semi-permeable membrane. So which means particles of water are now going to pass this semi-permeable membrane. So this means after some time we are going to see the change in the level of the solutions. This means now this level of sugar solution, because now is going to
receive some water molecules, it means this solution is going to rise. This solution is going to rise because water is going to move inside this solution. And here, the level of water is going to decrease, which shows that osmosis is now going to take place. And this water now is going to move inside the salt solution. So which means now the solution, water is going to move in here, and then the level of water is going to decrease, whereas the level of salt solution is going to increase. So now I would like to do a little bit theoretical experiment whereby we are going to use potato chips and this water and pure water. So now we can have two potato chips. First of all, we are going to have potato chip, we are going to put it in pure water. And now we are going to have another one. We are going to put it in salty water. So normally, for this experiment to work well, it means now, before putting these potatoes inside the water, what has to happen is you peel the potato. After peeling the potato or after cutting them like this, you take a scale, you find the mass of those potatoes. You take the scale and then you find the mass of that potatoes. And then you also take a ruler. You take a ruler, you measure the length of the potato. You measure the length of the potato using your ruler. Now, you can begin your experiment. You put one potato chip, you put it in pure water, and the other one you put it in salt water. Then you wait for some time. You can wait for about 30 minutes. Then, after then, there are some differences. There is something that you are going to see after we have done this experiment. What has to happen is the potato that was in pure water, after you remove it from pure water, you put it back on the scale. You are going to see the difference in masses. This potato is going to shrink a bit. Suppose the potato was 8 grams at first. When it comes from pure water, you might find out that it's now 7.5 grams. Why? It means now the potato, water moved out of the potato and escaped into the container. And now, by that, it means now here, osmosis have taken place. So it means now, water potential, a potato has water inside. As you know that plants, they have the waters and their mineral salts. So now it means the potato has lost its water to the water inside the beaker. So now, if you go into that potato that has been placed in salt water, we are going to see the opposite. This one, suppose our potato, here it was also 8 grams. You will find out that after you take it out of that salt water, you might find that the potato is now 9 grams, which means water inside the beaker moved inside the potato, which means now water moved inside the potato. So when water moves inside the potato is now going to gain size. So which means the length here of the potato, if it lost some mass, now it means that also the length is going to change a bit. If the potato here was about 5 centimeters, you will find out that now, because it has shrunk, maybe the length is going to be 4.5 centimeters. Here, if the potato was 5 centimeters, you will find out that now, because the potato has absorbed some water, it has gained some water, is now bigger in size. It means the length is also going to be altered. It might be 5.5 centimeters. So in the next lesson, what we are going to do is that we are going to talk about the plasmolysis and fluxidity. Because now, when a potato shrinks and then it loses some water, it means there is something that has happened. So we'll go into deeper details, and then even this experiment, we are going to do it practically together so that we can see all these things happening. Thank you.